Genetic cloning reaches a biological limit. 20-year study reveals mutational meltdown. A landmark 20-year study in Japan has demonstrated that mammalian cloning, when repeated over generations, inevitably leads to genetic degradation and ultimately extinction. The research, published in peer-reviewed journals, provides the first conclusive evidence of a genetic dead end in serial cloning, reinforcing the fundamental importance of sexual reproduction for species survival. The experiment, cloning mice through generations beginning in 2005, researchers at the University of Yamanashi repeatedly cloned a single female mouse, transferring her nuclear DNA into denucleated eggs for 57 successive generations. This created over 1,200 mice derived from one original donor. Initially, the process appeared remarkably efficient. Cloning success rates even improved with each generation. However, by the 58th generation, the recloned mice died within 24 hours of birth due to the overwhelming accumulation of genetic mutations. Muller's ratchet and mutational meltdown. The study confirms Muller's ratchet theory, which predicts that asexual reproduction, like continuous cloning, allows harmful mutations to build up over time. Unlike species with sexual reproduction, which can purge these mutations through genetic mixing, clonal lines suffer an irreversible decline in fitness. This is known as mutational meltdown, a point where genetic defects overwhelm the organism's ability to survive. Why this matters? For decades, cloning has been touted as a potential tool for conservation, pet preservation, and even human reproduction. This research does not invalidate short-term cloning applications, but it proves that long-term species survival cannot rely on cloning alone. The idea of recreating extinct animals through cloning alone is biologically unsustainable. The role of chromosomal abnormalities. The decline wasn't immediate. For the first 25 generations, the cloned mice remained healthy. However, after that point, the frequency of chromosomal abnormalities and coding mutations nearly doubled. The loss of the X chromosome became particularly problematic, but even earlier mutations didn't kill the mice outright. They simply accumulated until the 58th generation, when the system collapsed. Sexual reproduction as a genetic reset to test whether sexual reproduction could recover the degraded genomes. The researchers bred females from the 20th, 50th, and 55th generations with normal males, while older generations, 50th and 55th, produced smaller litters. Subsequent generations bred with normal mice recovered normal litter sizes. This proves that sexual reproduction can partially correct the damage caused by excessive cloning. But the longer the clonal line, the harder the recovery. The findings reaffirm the evolutionary inevitability that sexual reproduction is indispensable for the long-term survival of mammalian species. The authors conclude, this research doesn't dismiss the short-term benefits of cloning. However, it serves as a stark reminder that the natural processes of genetic diversity, driven by sexual reproduction, are essential for any species to persist beyond a limited number of generations.